Today we need to talk about enzymes. Enzymes are also known as helper proteins. They're a special group of proteins whose job it is to speed up chemical reactions so that life can exist. Without enzymes, there would be no life. They are known as biological catalysts. Biological meaning part of life, catalyst meaning to speed up. Everything you do is based on chemical reactions. When you eat something, chemical reactions are occurring to help you break down that food and get the molecules out of it that you need. When you breathe, chemical reactions are occurring in your lungs and bloodstream to get oxygen in and carbon dioxide out. So how important are enzymes? There would be no life without enzymes because chemical reactions occur very, very slowly on their own. But with enzymes, they happen very quickly. Without enzymes, when we eat food, we would starve to death before we were able to digest it. When our body produces waste, without enzymes, we wouldn't be able to get rid of that waste fast enough, and we would poison ourselves. We have two major groups of enzymes. We have synthesis enzymes, which means to put together or to build. We have digestive enzymes, which break down or take apart molecules. These reactions would occur naturally, but at such slow speeds that we wouldn't be able to live. So enzymes come in and they help out and they cause these reactions to occur faster so that life can continue. How do enzymes speed up reactions? They do so by lowering the activation energy. The activation energy is the amount of energy that is required for a reaction to occur. So for example, if a reaction requires 50 calories of energy to happen without enzymes, if an enzyme comes in and helps out, it would only require, say, 20 calories of energy. So now this reaction can occur faster because the body doesn't have to spend as much time building up the required energy because it has to build up to a lower level. The important thing to remember is that enzymes are proteins. And remember, the most important thing about a protein is its shape. So enzymes don't run around interacting with every single molecule they bump into. There is a specific enzyme to react with a specific molecule. So you have hundreds and thousands of different enzymes, each one with a specific job. Because of its shape, it can fit in with its partner perfectly, like a puzzle piece or like a lock and a key. They only work with each other and nobody else. Enzymes are named for the reaction that they work with. So we have a nice new naming rule to help you out. Remember, with sugars, they always ended in O's. Well, enzymes are always going to end in A's. So for example, sucrase breaks down sucrose. A's lets you know it's an enzyme. Suc lets you know it works with sucrose. Proteases break down proteins. Lipases break down lipids. And DNA polymerase builds DNA. So they all end in A's. So that's your nice new naming rule to help you out. A really important part of an enzyme is the fact that it doesn't get used up in the reaction. It's not part of it. It doesn't change its identity. It just comes in, helps, and leaves. This is really important because it means you only need a little bit of enzyme to do a lot of work. Let's think of an analogy to help us understand this more. Let's say I'm moving. So I packed all my stuff up into 50 boxes. I have 50 boxes that I need to move onto the truck. Now, if I invite over some friends to help me, let's pretend my friends are the enzymes. If one friend comes over and moves one box and then says, that's it, that's all I can do, I'm all used up, well then 50 boxes, I'm going to need 50 friends. Going to have to buy a lot of pizza to pay them back. But like enzymes, my friends are not used up in the reaction. So that means that one friend can move one box onto the truck and then come back and pick up another box and move onto the truck. So now instead of needing 50 friends, each to move one box, I only need to invite over 2 or 3 friends and they can move multiple boxes. Same idea with enzymes. They don't work with one molecule and then they're done. They work with their one molecule and then they can go find another molecule just like it and do the exact same reaction with it over and over and over again. So you only need a little bit of enzyme to do a lot of work. If this doesn't make sense to you, please pause the video here and raise your hand and ask for help from me, because this is a really important concept that you need to understand. Now let's talk about some vocabulary. Let's look at the image below representing an enzyme doing its job. This is an important tool that you're going to need to be able to use. You need to be able to label an image similar to this. So first we need to decide which one of these molecules in the image is the enzyme. Well, we know that the enzyme isn't used up or changed in the reaction, so it's going to look the same going in as it does coming out. So if we look at our image, that's going to be the purple blob, okay? Because going in on the left looks the exact same coming out on the right. It wasn't used or changed. So the purple blob is the enzyme. All enzymes have a very special area, which is the perfect shape to fit with its particular molecule. That area is called the active site. The particular molecule that the enzyme is capable of working with and fitting perfectly with is called its substrate. When the substrate and the enzyme lock together in the active site, we call it the complex. Sometimes it'll be referred to as the substrate-enzyme complex. After the enzyme has done its job, either putting together or taking apart the molecule, it releases the changed molecule, and this is called the product. So in the picture, notice one yellow molecule goes in, 2 different things, the products, come out. So an example in real life is an enzyme known as peroxidase. So the purple blob would be peroxidase. Peroxidase lives in our liver, and it breaks down hydrogen peroxide, which is a waste product that our body produces and would poison us if we didn't get rid of it. So if we imagine that this yellow block is the substrate, that would be hydrogen peroxide. When the 2 come together in the complex, the hydrogen peroxide is bonded with the peroxidase enzyme. The enzyme does its job, breaks the hydrogen peroxide into oxygen and water, the products. Oxygen and water are not poisonous to us. So now we have taken something poisonous and turned it into something useful. Hydrogen, or oxygen, and water. In case you didn't write down the definitions on the last screen as I was going over them, here are the vocabulary words that I just went over and their definitions. So pause this video as needed to be able to get those definitions. And remember, you need to be able to label that image. Sometimes you will hear the enzyme and its complex referred to as a lock and key model, and that is just referring to the fact that one enzyme will only react with one substrate because they fit together perfectly, like a lock and a key. Remember, the most important thing about a protein is its shape. So the most important thing about an enzyme is its shape. There are several things that can affect how well an enzyme does its job. One is its shape. If it's not the right shape, then it cannot interact with its particular substrate because it won't fit. Another thing that can affect an enzyme's Activity is concentration. How much enzyme is available for the reaction to occur? And lastly, temperature. Proteins, if the temperature gets too high, it falls apart. It denatures, so it will not work anymore. And finally, it's pH. Remember, proteins need a specific pH to function. Well, an enzyme is a protein, so enzymes need a particular pH for it to function appropriately. I'll give a little bit more information about the concentration, temperature, and pH. Why is concentration important? Well, as you increase the amount of enzyme, you're going to increase how fast the reaction can happen. Because the more enzymes you have, the more that they're interacting with their substrate and working. But this only works up to a certain point. Then you're going to reach what's called a plateau, or the fastest that it can occur. And then it doesn't matter if you keep adding more enzyme, it won't be able to work any faster because you're just basing it on how often the enzyme and the substrate can come into contact with each other. Optimum temperature. In humans, we have an optimum body temperature of 37 degrees. That's our average. So our enzymes work best at 35 to 40 degrees, right around our temperature. Now, if the temperature gets too high, you're going to denature your proteins, denature your enzymes. They're going to stop working, and that's really bad. That's why it's so important to get fevers under control so you don't have brain damage. Now, if you lower the temperature, what happens when you lower temperatures? Molecules move slower. Heat speeds things up. Cold makes it slower. So if you lower the temperature, your reactions are going to happen slower. And that's just because the enzyme and the substrate aren't going to come into contact as quickly. Now, pH changes a protein's shape. So if your protein gets into a situation where the pH is too low or too high, it's going to denature. And remember, that's permanent. Now, this is going to depend a little bit on where it is in the body, though. We have an enzyme in our stomachs called pepsin that helps us to digest. Now, in our stomach is very acidic. It's full of acid. That's why when you throw up, it burns your throat because it has acid in it. So pepsin works at a pH of 3 so that it can work inside your stomach. But if pepsin leaves the stomach for some reason and gets somewhere else in the body, it's going to denature because it only works in high acidity. Now, on the other hand, in our intestines, a little bit after the stomach, we have another enzyme known as trypsin. Now, it's going to work at a pH of 8, which is more basic because the inside of our intestines are at a more basic level. So trypsin is happy in the small intestines, but put trypsin in the stomach where it's an acid, and it's going to denature, and it's going to stop working. So, just a reminder, what is the most important thing about enzymes? Their shape.